Good afternoon, everyone. My name is Vasily Grinev. I'm Associate Professor of the Department of Genetics at the Faculty of Biology, Belarusian State University. Today we will talk about analysis and annotation of big genetic data. Since this lecture is introductory one, we shall confine ourselves to discussion only three questions. Genome assembly, identification of genetic variations in a genome, and analysis of gene expression. The genome assembly consists in the merging of short sequenced DNA fragments, called reads, to restore the original sequence of the genome. This is needed as a high-throughput DNA sequencing technologies cannot read whole genomes in one, but rather read small pieces of genomic DNA. There are two very different types of assembly. First, mapping to a reference genome, and second, de novo assembly without prior knowledge. Regardless of the type of assembly, we should assemble contigs at first, scaffolds at next, and whole genome at finally. A contig is a set of overlapping reads that together represent a consensus region of DNA. A contig is a continuous stretch of sequence containing only A, C, G, or T nucleotides, without gaps. Scaffold is created by chaining of the contigs together using additional information about the relative position and orientation of the contigs in the genome. Contigs in a scaffold are separated by gaps, which are designated by a variable number of n letters. In the case of assembly, by mapping to a reference genome, all reads are independently aligned against reference sequence. In general, alignment or mapping is a process of determining the most likely source within the reference genome sequence for the observed read, given the knowledge of which species the sequence has come from. So, to do this, we need a reference sequence and an efficient and accurate alignment algorithm. The sequence of the reference genome, which we are interested in, we can find in specialized database, for instance, Ensemble. There are two different types of alignment. When the alignment is global, the query sequence, read in our case or any another sequence in general case, must be aligned along its entire length. Alternatively, local alignment is aimed at finding a subsequence of query sequence with the best alignment against reference sequence. As for effective alignment algorithm, many different approaches to solving this problem have been developed. One approach is based on indexing of reads and the genome using hash tables. Align a subread is a good example of such algorithms. Another one is indexing genome using suffix array or Burow's-Wheeler transform, and the both time 2 is a representative of this kind of algorithms. And finally, some approaches use merge sorting, such as the slider algorithm. In fact, algorithms are based on indexing of reads and the genome using hash tables are most popular today. Basically, hash table includes two columns. The key column lists all the unique short oligonucleotides extracted from the reference sequence. And the value column stores the genomic coordinates or origin of these oligonucleotides. Usually all 16 mer oligonucleotides are extracted, for example, with a shift of 3 nucleotides from the reference sequence, and informative ones are stored in hash table. Armed with a hash table, now we can quickly determine the genomic origin of the read. We can use a seed and word based algorithm implemented in a linear subread to illustrate this situation. This algorithm extracts the unique informative oligonucleotides, called also subreads or short seeds, from the read of interest. The set of informative subreads is compared then with a hash table and quickly determines their most likely genomic origin. Since short 16-mer oligonucleotides can be mapped to different sites of the genome, the algorithm allows all subreads to vote for the optimal location of the read. After mapping, further assembling the aligned reads into contigs and scaffolds is a simpler task. Another approach is de novo genome assembly. This approach is aimed to assemble a new genome from scratch, without extra data and prior knowledge. The first step of the de novo genome assembly is to build an assembly graph from overlapping reads, or alternatively, de Broglie graph from reads-derived k-mers. The second step is to find unique paths in a graph to get a contigs. Each unique path or contig in a graph represents a unique sequence or fragment of a new genome, called consensus. At the third step, individual consensus sequences are combined into scaffolds and thus in turn into one total consensus representing the entire genome. The final step in de novo assembly is assessment of assembly quality. There are several criteria for such an assessment. For instance, total consensus should be close to the expected genome size. Or another criteria is the number of contigs must be as small as possible, and last criteria is N50 should be as much as possible. In this case, the N50 is the shortest scaffold at 50% of the genomic size, as shown at the bottom of this slide. If the genome is assembled, we can now look at the variability of the nucleotide sequence. In our discussion, we confine ourselves to one group of variability, which is called short sequence variations. This group of variability includes three types of variations. The first type is single nucleotide polymorphism, or simple SNP. SNPs are the most common sequence variations at single nucleotide level that occur approximately once every 100 to 300 nucleotides in genome and less 1% in human populations. The second one is insertion-deletion polymorphism, or indels. In fact, indels are small multinucleotide deletions or insertions in the genome. And finally, the third type is short tandem repeats, or simply STRs. STRs are represented by a different number of repetitions of the same short nucleotide sequence as, for example, the CTS sequence in the case presented on the slide. To find a variable site, we need to compare the assembled genomes or aligned mapped reads with the reference genome. Any mismatch in the nucleotide sequence of these two genomes may indicate the presence of variability as illustrated on the slide. Here we compared reference genome, any part of specific gene, with aligned reads, and this site is possible SNP site. However, in addition to the true variability of the genome, there are several possible reasons for the discrepancy. In particular, the mismatch may be due to an error generated during sequencing library preparation, a nucleotide calling error, misalignment, I mean error in mapping, or an error in the reference genome sequence. To date, many approaches have been developed to establish the true variability of the nucleotide sequence. For example, the approach based on the binomial model of mismatch distribution is quite popular. And our last topic is the analysis of gene expression. Gene expression is a process by which information recorded in a gene is used to the synthesis of gene product. The first stage in this process is always the synthesis of RNA molecules. Therefore, we can estimate the level of gene expression by the number of synthesized RNA molecules. With RNA-seq data, the basic workflow of gene expression analysis includes several steps. The first step is summarization of aligned reads at gene level and development a count matrix. This step can be extended by filtering a count matrix against low sequencing depths to reduce noise in the data. The second step is estimation of the normalization or scaling factors to normalize RNA-seq libraries of different size, for example, with different total number of reads. This step is important when we carry out a comparative analysis of gene expression and use several biological samples. In this case, each sample usually has a different total number of reads and, as a result, a different sequencing depth of genes. There are various approaches to calculating the normalization factors. For instance, the normalization factor for each RNA-seq library may be estimated robustly using the trimmed mean of m-values or TMM normalization method. By this method, the normalization factor for the RNA-seq library K using the reference library R is calculated as shown on the slide. Normalization factors across several samples can be calculated by selecting one sample as a reference and calculating the normalization factor for each non-reference sample. You may find more detailed information on this method of normalization in the work of Robbins and Oshlag, a scaling normalization method for differential expression analysis of RNA-seq data, published in 2010 in Genome Biology. The third step in gene expression analysis is stabilization of the data variability. In fact, RNA-seq data show some degree of heteroscedasticity. This feature of the RNA-seq data is clearly visible on standard pairwise scatter plots or on plots of mean-variance relationships. The problem of heteroscedasticity is further aggravated by the fact that, as a rule, it's difficult to find a plausible model of RNA-seq data distribution due to a limited number of available biological samples or biological replicates. Moreover, raw biological data, including RNA-seq data as well, usually can be described with a raw distribution model other than a normal distribution. One of the most effective solutions in the problem of the RNA-seq data variability was proposed by Law and colleagues in the method called VUM. It's mean variance modeling at the observation level. The VUM method estimates the mean variance relationship of the log2 counts, generates a precision weight for each observation, and enters this into subsequent empirical based analysis pipeline. When the weights are incorporated into a linear modeling procedure, the mean variance relationship in the log2 counts is effectively eliminated. You may find more detailed information on this method in the work of Law and colleagues, VUM, precision weights, on log linear model analysis tools for RNA-seq read counts, published in 2014 in Genome Biology. So, now we have log2 transformed and normalized relative to effective size of each RNA-seq library counts for each gene with respective weights. We call these counts as log2 CPM, logarithmically transformed counts per 1 million of counts in RNA-seq library. The weights are then used in the linear modeling process to adjust for heteroscedasticity. Next, we consider two scenarios. First, we need only a list of genes that are expressed in the biological sample of interest. And second one is we want to compare the expression of genes in two different conditions, for example, normal versus disease. In the first case, it's sufficient to select the appropriate threshold and select those genes for which the log2 CPM value is above the selected threshold. The second scenario is more complex and is implemented using linear modeling. The linear model is a general frame for modeling and data analysis in statistics. It consists of defining a linear relation between observed values and experimental conditions. Let this vector is the response vector for the GTS gene. Here, the response vector means a vector of transformed and normalized counts that support expression of GTS gene. Assume a simple linear model like this equation, where X is a design matrix and Aj is a vector of unknown coefficients to be estimated. In the simplest two-group comparison, disease versus normal and n equal to 3 per group, the design matrix can be represented as shown in the slide. You may see the very simple design matrix. To obtain unknown coefficients Aj, the above linear model is fitted by linear regression with least squares to the log2 CPM values for each gene. Now we can find the difference between experimental conditions. Define contrasts of the fitted coefficients as shown in this slide. In our simplest two groups comparison, disease versus normal, we will estimate one contrast, and the contrast matrix can be represented as shown in the slide. If we apply this contrast matrix to fitted linear models, we will return a model object recalculated in terms of the contrasts. In this new model object, the coefficients are logarithms with base 2 of fold changes, or log2 fold change, or simply log FC between the experimental conditions. And our final question is, do calculated log FC coefficients significantly different from 0? Unfortunately, we cannot use traditional statistics to solve this problem because, first, RNA-seq data can be described with other statistical model than a normal distribution, or we are unable to establish an exact shape of distribution. Second, RNA-seq data includes usually small number of replicates n and large number of assessed genomic features f, genes in our case. Third, small n causes poor estimations of the variance. Fourth, large f leads to identification of features with very small variance by chance. And last, fifth, the top-ranked list will be dominated by the features with extremely small variance. Together, above-mentioned points lead to high false positives. Empirical Bayesian method is one of the current solutions of this problem. It was firstly proposed by Ingrid Lundstad and Terry Speed in 2002 for two-colored microarray data. This approach was subsequently generalized by Gordon Smith in 2004 to a linear model framework. Smith's method can be applied to both single-channel and two-colored arrays as well as to RNA-seq data. You may find more detailed information on this method in the work Smith's J.K. Linear Models and Empirical Bayes Method to Assessing Differential Expression in Microarray Experiments, published in 2004 in Statistical Applications in Genetics and Molecular Biology. So, today we have discussed three questions concerning the analysis and annotation of big genetic data, since assembly of genomes from short reads to analysis of gene expression, including differential expression of genes. Of course, our discussion was limited to the conceptual level, and many details were not considered. All additional information can be found in various publications, which are mostly freely available. I wish you success in this matter. Good luck!